I'm Kyle Medak, a postdoc in Daniel Drucker's lab at the Lunenfeld Tannenbaum Research Institute at the University of Toronto. I'm excited to give you a quick overview of our research paper published in JCI Insight, showing how semaglutide reduces murine blood pressure through vascular smooth muscle GLP-1 receptors. This paper is the hard work of various members of the Drucker lab, as well as our collaborators at Boston University School of Medicine. GLP-1 receptor agonists, aside from their effects on blood glucose and body weight, have shown cardiorenal benefits to lower cardiac events and blood pressure. More recently, in 2025, the FLOW trial has demonstrated kidney benefits, but more work is needed to determine how. It's been shown by our group that benefits to the heart don't rely on heart GLP-1 receptors, so we hypothesize that actions on the kidney can induce the observed benefits to the heart and kidney and are related to hemodynamic effects to lower blood pressure. This is because we observe GLP-1 receptors in vascular smooth muscle of the kidney. To demonstrate which kidney cells express the GLP-1 receptor, we analyzed a comprehensive dataset of mouse single-cell RNA sequencing. We show here that vascular smooth muscle cells are displayed in pink on the left-hand side, and when we highlight the GLP-1 receptor expression on the right, we observe the signal for GLP-1 receptor in vascular smooth muscle cells. We also highlighted vascular smooth muscle cell marker MYH11, shown here on the left. The expression of this marker overlaps with the GLP-1 receptor expression in the kidney, telling us that vascular smooth muscle is a likely candidate for kidney GLP-1 receptor signaling. Thus, we utilized MYH11 as a genetic driver for conditional knockout of the GLP-1 receptor in vascular smooth muscle cells using Cre recombinase. When we induce this VSM knockout with tamoxifen, we observe a greater than 95% knockdown in whole kidney homogenate and in renal artery, confirming that renal GLP-1 receptor expression is mainly in vascular smooth muscle cells. In kidney histological analyses, staining with a validated GLP-1 receptor antibody, shown here in green, overlaps with smooth muscle cell actin, which stains the vasculature in red. This signal is lost in the kidney of our VSMC GLP-1R knockout mice. These are evidence that the GLP-1 receptor is localized to the vasculature of the kidney in VSMCs. When comparing wild-type and vascular smooth muscle GLP-1R knockout mice, we found no differences in body weight, glucose tolerance, blood pressure, and no difference in the mRNA expression of the GLP-1 receptor in other tissues which express GLP-1, such as the aorta, heart, and the phenotypic difference we do observe in these mice is in acute blood pressure lowering effects of semaglutide, which is lost in vascular smooth muscle cell knockout of the GLP-1 receptor. We measured blood pressure using the tail cuff CODA blood pressure system and repeated these findings under conditions of hypertension. We used three different modalities to induce hypertension in our mice: L-name in the drinking water, overexpression of renin with adenovirus, and angiotensin II mini-pump implantation. In all of these hypertensive modalities, semaglutide acutely lowered blood pressure in wild-type animals, while this effect was lost in knockout animals. This is evidence that blood pressure lowering effects of the GLP-1 receptor agonists rely on vascular smooth muscle GLP-1R signaling. To test if repeated treatment would result in sustained blood pressure reduction, we treated mice for 3 weeks with semaglutide, during which hypertension was induced by L-name in the drinking water, and after these 3 weeks, acute treatment still reduces blood pressure in wild-type mice. In line with this, left ventricle mass and whole heart weight are reduced in these mice, providing evidence that each treatment with semaglutide results in reduced blood pressure, and these exert cardiac benefits during hypertension. Alternative theories have been proposed for the underlying mechanisms of blood pressure reduction by GLP-1 receptor agonists, and they might be attributed to endothelial cell GLP-1 receptor signaling. To test this, we studied an endothelial and immune GLP-1 receptor knockout using a Tie2 Cre promoter. We hypothesized that if endothelial cells are essential for blood pressure regulation, then we would lose the blood pressure effect of semaglutide in these mice. Interestingly, semaglutide still lowered blood pressure in endothelial cell knockout mice, reinforcing that it is likely vascular smooth muscle cells that regulate blood pressure effects of GLP-1 receptor agonists. To give insight on how vascular smooth muscle cells might regulate blood pressure, we measured urine volume. Semaglutide increased urine volume in both wild-type and knockout animals. Similarly, urine sodium and chloride are increased in wild-type mice and to a lesser extent in knockout animals. In contrast, Glomerular filtration rate is increased by semaglutide in control animals, and this effect is fully lost with vascular smooth muscle cell knockout of GLP-1 receptor. This is in line with increased blood flow shown by our collaborators at Boston University, who analyzed blood vessels ex vivo, and after preconstriction, semaglutide promoted relaxation measured here in mesenteric artery. Lastly, we analyzed proteomic changes induced by semaglutide in the kidney and renal artery. Here in the kidney, semaglutide produced bidirectional changes to pathways related to platelet aggregation, clot formation, lipid metabolism, and apoptotic signaling. These regulated proteins, shown here in red and blue in the volcano plot, will now be labeled in yellow when analyzing knockout animals. None of the proteins are changed by semaglutide anymore when GLP-1 receptor is knocked out of the vascular smooth muscle cells. We can see this represented visually using a heat map. In the leftmost columns, semaglutide regulates these proteins shown in red. On the right side, this relationship is lost in knockout animals. Altogether, we're showing that GLP-1 receptor agonists have direct effects on the kidney. The GLP-1 receptors are in vascular smooth muscle cells, and they relax blood vessels. They induce diuresis and natriuresis and proteomic changes. We know that weight loss, diuresis, and natriuresis undoubtedly have impacts on hypertension over the long term. But our acute experiments show that semaglutide has effects on blood pressure independent of these factors. Quickly after treatment, vascular smooth muscle cells promote vascular relaxation. This is important for lowering blood pressure, an effect that is still potent after weeks of treatment.